Unless you've been living under a rock, you'll agree that AI is being explored, used, and abused across industries. This includes natural sciences and pharmaceutical research and development, where we've seen frenzies of investments and bold predictions. But are drugs that are discovered or otherwise touched by AI actually better? In this video, we'll review broader evidence in the field and also a specific case example of the most advanced drug discovered and designed by AI. Let's first address the elephants in the room for many viewers and students: AI capabilities and risks. AI models are becoming more than decent in theoretical chemistry, even if they might not be particularly helpful for your total synthesis homework. Just as we've seen in other exams like the BAR or LSAT, the performance in standardized chemistry tests is steadily increasing across models. In curated chemistry benchmarks, LLMs also seem to consistently outperform the best experienced chemists. We obviously have the annoying hallucinations and overconfidence, and there is a difference between large language model outputs and true understanding or creativity. But still, this performance is pretty impressive and will only increase over time. This begs the next question: will AI steal our jobs? This depends on how easily your job might be automated or replaced with generative AI mumbo jumbo. Here's a somewhat comforting quote from the founder of an AI biotech company that we will talk about later. This topic could be a whole video by itself, and everyone has their own opinions. If you believe the hype out there, you would think that AI is literally curing cancer already, and even experimental chemists will soon be replaced by robos and automated synthesizers. What do you think? How will things look like in 10 years? Let me know your predictions in the comments. Amidst the frenzy of magical AI, there are some companies resisting the hype. After all, a lot of the technology and exploratory use cases have been around for years already. But these are rare exceptions, as most companies are trying to pimp their corporate narratives and financing prospects. This is of course happening across all industries, but AI seems especially promising and hot in drug development. But why? Most of you will know that finding a new medicine is trying to find a needle in the haystack. In early research, scientists have to sift through thousands of molecules to identify drug candidates which have the highest probability of being efficacious and safe. This process already takes years and millions in funding, and unfortunately, that's the easy part. Once we have what we believe is the needle, we have to put it through 3 phases of clinical trials in humans, where only 1 in 10 candidates make it max. In 90% of the cases, we put in time and resources only to find out that the drug did not result in statistically significant benefits, or that it's not as safe or tolerable as we hoped. The bar for new drugs is also increasing because we found hundreds of great medicines over the last decades. We get less and less medicines per dollar invested because we need to find new and better drugs. This is where pharmaceuticals differ from, let's say, the entertainment industry, where consumers will gladly watch the 15th copy-paste superhero movie. In contrast, no one will pay for an undifferentiated medicine if we have cheaper alternatives or generics or biosimilars on the market already. Not only is finding drugs more expensive, the already long process is getting longer and longer over time. This is quite the double whammy. One reason is that it simply takes longer to find new patients because, as mentioned, we have decent alternatives in many diseases already. Another reason is drugs need to show more and more information and data, so clinical studies are becoming more complex. More complexity means more measurements, time and money needed, while still having less than 10% success rate in the clinic. All of this could be sustainable if financial returns on the risky bets and investments would at least be similar. However, the sales of new drugs also tend to decrease, again because of available alternatives, pricing pressures, and the fact that many new drugs help smaller patient populations. Because drugs are not magically created out of thin air, companies are investing more than $200 billion in their pipelines globally, and these R&D dollars need to come from somewhere. Even more urgency is added because the industry is heading towards a large patent cliff, so it would be very welcome if drugs could be developed more cheaply or quickly. Overall, we need to put in more, but we're getting out less. Because drug development is based on hundreds of individual steps with high complexity, the first hope for AI is that it will help us to go through these steps with higher speed and efficiency. This is more operational, so doing the same thing with less resources or time. The bigger hope is the buzzword of transformational impact, where AI might help us take smarter decisions, uncover new discoveries, and find better drugs than we could without it. On the operational side of things, one relevant example is the automation of synthesis planning. This has been a long-standing effort in chemistry well before the recent hype, but it's now becoming more common to use machine learning-based tools for retrosynthesis and also the prediction of reaction outcomes. The number of possible disconnections in a synthesis can be very high, so retrosynthesis programs need to evaluate, rank, and optimize different approaches by number of steps, use of protecting groups, or costs of starting materials. Easier synthesis lowers costs and accelerates real-world testing of compounds, so the hope is that we can find promising drug candidates more quickly. A more transformational but less clear example is the prediction of drug properties, like safety. Medicinal chemists already know many functional groups that are often associated with toxicity in humans. Using these structural alerts is very common in drug design, but simply categorically avoiding all of these functional groups will also outlaw many good drugs. Almost every molecule has some safety risk behind it, so it all depends on the dose, the speed of drug metabolism in the body, and other factors. Here the idea is that a smart AI could somewhat accurately predict whether a given drug would be toxic or maybe trigger certain side effects. Such safety predictions would be essential for scientists to phase out animal testing, which the FDA has recently announced it plans to do. This will surely take longer than the 3 to 5 years claimed here and depend on other tools like organ chip models, but we see that there's scientific and ethical value of this more transformational AI use case. So are drugs discovered with the help of AI really different? An analyst paper from last year reviewed the drug pipelines of so-called AI native biotechs, so small drug development companies specializing in AI. As you would expect, the baseline number of molecules which were discovered or otherwise touched by AI increased over the last years, with almost 70 being in ongoing clinical trials as of 2023. The top chart shows the distribution by clinical trial phase, and the bottom chart shows the broad mode of AI discovery. So, if it was used to find the target or if it was used to design the actual drug. The obvious question is, what makes a company truly AI native and what qualifies as an AI discovered molecule? You can check out the supplementary information for details, but obviously a lot of this relies on selective information released by companies that probably want to overplay their use of AI and seem as innovative and hot as possible. You could also debate many of the classifications by the authors because some of the drug targets claimed to be discovered by AI were already well known. So there's a few real novelties and a bunch of fake ones as well. What about success of these AI projects? Where clinical data is available, the authors found that success rates in phase 1 were much higher than historic averages observed for all non-AI discovered drugs developed by industry. In phase 2, probability of success seems more comparable with the general benchmarks. Remember that phase 1 trials are small studies that focus on safety, while phase 2 trials are a bit larger and investigate the efficacy of the drug. You could look at the differences in success rates based on two angles. The pessimistic view is that the sample size of the review is way too small to make any conclusions. Also, given the existing data that the models are based on, many AI-derived molecules might go after well-known biology or might be structurally similar to known compounds. So there is a de-risking of potential toxicity, and thus it is not surprising that phase 1 trials see more success on average. Also, remember that success here just means progression from one phase to the next. So it could be that AI native biotechs are better funded and have a higher risk appetite to even progress molecules with suboptimal phase 1 data into phase 2 nevertheless. In contrast, the optimistic offers suggest that higher success might be because AI algorithms are already able to select drug-like molecules with above-average safety or metabolic profiles. The utopian breakthrough would be that AI could even predict efficacy of drugs, but based on the comparable success rates in phase 2, we're clearly not there yet. Beware that this whole analysis was done by consultants who might have an interest in selling AI-related projects to clients, so they might need to hype things up a bit. Within this wave of new AI-discovered drugs, the molecule right here is among the most advanced. Its developer In Silico Medicine recently made headlines after releasing supposedly very promising data from its phase 2A clinical trial in a lung disease. The discovery story behind it is indeed pretty cool. In a first step, consolidation of different datasets and scoring of associations between them suggested a new hypothesis related to the protein TNIC. This isn't a completely new target, as it was investigated in the past to treat solid tumors, but unfortunately without success. The new insight from AI was that the TNIC pathway might play a role in the fibrosis or scarring of lung tissue in IPF. This is a progressive lung disease with poor prognosis and limited treatment options, so quite different than cancer and actually innovative. After this target identification, the second step also used generative AI to suggest potential drug structures that optimize target binding and other properties. Importantly, the scientists still had to synthesize and test thus because the TNIC target was already known. The optimization started from crystal structures of known inhibitors. If you compare the molecules, you'll see that the AI-designed drug has a unique structure, so it is quite the real optimization and not just minor changes based on a copycat. Now, before we get into how this compound was synthesized and how promising it is, we need to admit that we've participated in the buzzword bingo and threw the term AI around like confetti at a party. But do you know what AI actually is and how the models truly work? I'm not saying we will all receive million-dollar pay packages if we do, but all of us can take steps to better understand and apply AI for our own benefit. The perfect way to do this is through Brilliant, an interactive learning platform with thousands of lessons that exercise your brain and help you become a better thinker. We've previously highlighted Brilliant's insightful lessons like the one on quantum mechanics and computing, but Brilliant's broad content library also includes courses on neural networks and AI. No worries if you're not an expert coder. With the help of hands-on lessons and examples, you will quickly learn how generative AI models work, how they're trained, and even how to build a large language model yourself. Beyond AI, you can also get smarter on math, science, programming, and data analysis. Simply pick your favorite area and enjoy Brilliant's unique mix of engaging problems, detailed explanations, and daily encouragements to stay motivated and establish consistent learning habits. To try everything Brilliant has to offer for free for a full 30 days, visit brilliant.org/totalsynthesis or scan the QR code on screen. Or you can click on the link in the description. You will also get 20% off an annual premium subscription. Before we review the clinical data of this most advanced AI-discovered drug, we can review some organic chemistry as well by checking out how this compound was synthesized. It looks like a traditional pharmaceutical, and such structures with aromatic rings and amides are based on coupling reactions of smaller rings and fragments. In this case, 3. The amine on the left will be used to form the amide, so the question is really how we make the 2 other fragments. In our imidazole ring, the strange SEM abbreviation represents a protecting group that is a bit like a strange cousin of the better known MOM group. It's introduced at the very beginning and useful to suppress side reactions of the nucleophilic amine in imidazoles and pyrols. If you haven't seen this group yet, you might want to already think about how it can be deprotected later on. As a next step, the bromide was introduced through aromatic bromination with the use of catalytic AIBN as the radical initiator. Any undergrad student should know this mechanism very well. The regioselectivity stems from the fact that the CH bond between the two nitrogens is the weakest and thus most activated. This new bromide will be used to connect the intermediate with our second fragment. To make that one, we can use a Suzuki coupling of another brominated imidazole and a boronic acid which comes with the fluoride substituent. As just one interesting insight from the structure-activity relationship studies, this fluoride proved to be quite essential. Replacing it with a trifluoromethyl or a chloride group reduced activity at TNIC significantly, probably because these larger groups cannot fit as nicely into the pocket of the protein. As a next step, the free amine is again functionalized, but this time it's not a protecting group, but rather a simple isopropyl which is needed in the product. To create the cross-coupling precursor, the chemists first introduced an iodine and then swapped it out by lithiation and addition of tributyl tin chloride. The iodination is an electrophilic aromatic substitution, so the regioselectivity is now different compared to the prior bromination and instead driven by stabilization of the positive charge. Next, the two fragments were connected through a Stille coupling and the ester group was hydrolyzed and converted to the amide with the aid of an activating agent. The last step was SEM deprotection with strong acid, basically dissolving the compound in TFA. To advanced viewers, the mechanism is basically a mix of a MOM and a TROC deprotection, as we have protonation of the ether oxygen followed by beta elimination of the silyl group releasing ethylene. Aqueous basic workup converts the hemiaminal into the free amine and we get our target molecule Rentosertib. But is this compound a promising drug? Prior phase 1 trials already suggested an acceptable safety profile, and this phase 2a trial was now an opportunity to explore whether inhibition of TNIC improves lung function of patients with IPF. To measure this, the trial looked at changes in so-called forced vital capacity. This is the volume of air that can be maximally exhaled out of the lungs after taking a maximally deep breath. The higher the FVC or the less it deteriorates over time, the better the lung function. This data compares the change of FVC at 12 weeks of treatment versus baseline at week 0. You can pause the video here for a moment. How convincing does this look to you? The authors put a positive spin on it, noting a trend towards an increase with the 60 mg dose. We don't have the p-values, but the major overlap between the two confidence intervals indirectly gives us the conclusion. None of the groups had a statistically significant improvement. The pure numbers indicate that lung function worsened in the placebo group while improving in the 60 mg group. But we can't tell whether this is a random effect or truly a benefit of the drug. The sample size of patients is simply too small. One of the key question marks in this study is that a high share of patients missed their FVC measurements. The authors tried to perform some statistical magic to impute missing values, but we're not going to look at that data because it's not worth a lot and doesn't change the story. One additional argument was made when differentiating patients based on their background therapy, so ones that received standard of care medicine on top of the investigational drug versus those who did not. Again, feel free to pause and think about whether this looks any more convincing. With these subgroups, you might argue that for the no standard of care on the left, the FVC improvements seem more cleanly separated. This is a statistically significant change on paper, but again, pay attention to the N. We're talking about just 4 versus 9 patients here, which is an extremely exploratory comparison. The takeaway is that we really cannot tell at all whether this molecule has any positive, neutral or negative effect on lung function. 12 weeks is also pretty early, so you'd optimally want to know in future studies how this endpoint looks like after 6 or 12 months. The primary objective of this small study was to look at safety and tolerability of the drug, and here again, online commentary suggests a decent profile. This is also a bit more debatable because quite a lot of patients discontinued treatment and dropped out of the trial. Most of them did so due to adverse events with liver injury or dysfunction emerging in some patients. Some of these might have been driven by the existing standard of care background therapy, but the profile isn't really a home run. The trial also reported pharmacokinetic measurements, but even those raised some questions. One of them was the area under curve, which quantifies the total exposure to a drug. The other measurement is the Cmax, which is the maximal concentration that the drug reaches in a patient's blood. As you would expect, both metrics increased at the highest 60 mg dose, but there was a high variability. If we look at the Cmax, one patient seemed to have almost inexistent exposure, but even excluding that, there was a factor of 12x between lowest and the highest values. The authors didn't comment on this, but the PK profile also seems quite complex. As a final limitation, the patient population was very homogeneous given all participants were residents of China. This argument typically surfaces if China-only studies are positive, but because the data at hand isn't incredibly promising, this limitation doesn't matter too much. You really have to ask yourself if there would be as much enthusiasm for this data if AI would not be in play. Probably not. To further interrogate the potential efficacy signal, the company is evaluating next steps, and another phase 2A trial is already ongoing in the US. The sample size will be even smaller, so it will be interesting to see whether there are any differences to the China-only trial. Quite a few other clinical stage drugs were recently terminated by another key AI-native biotech company. Some molecules due to weak data, others due to quote-unquote strategic reprioritization. It's quite clear that AI is not a magic bullet, yet. What we can say with certainty is that many more AI-enabled drugs will enter clinical trials over the next months and years, providing the desperately needed sample size to reach any conclusions. I hope we'll all be around in a few years again to reflect on this question together. For the sake of patients and thus all of us, I surely hope that we will see some true breakthroughs in this field in the future. What do you think about all of this? If you've enjoyed this video, let me know with a comment or a like, or consider subscribing if you haven't already. A big thanks goes out to all my channel supporters. The process of making these videos isn't easy and often quite annoying, so your support is highly appreciated. Thanks for watching, and as always, until next time.